Hi everyone, thanks for joining us today for our In the Lab webinar with Dr. Sabrina Sofia Burgener. I'm Julie Chmielowski, I'm the Cell Analysis Program Director at Millennium Science, and I will be your MC today. Before we get started, a few housekeeping items to announce. So for everyone joining us today, you will go into the draw to win a $50 Amazon voucher. The prize will be drawn after today's webinar, and the lucky winner will be announced via email. So keep an eye on your inbox. Should you have any questions for our speaker today, please feel free to post them in the chat as we go, or hold on to your questions for the Q&A session at the end of the presentation. Our mission at Millennium Science is to ignite life science research and discovery with revolutionary technologies, and we partner with manufacturers that are market leaders in their field. We are excited to be supporting Agilent Technologies with today's webinar. When it comes to gaining deeper insights into cell biology, it's all about finding the right tool to solve your research problem. Agilent's cell analysis portfolio gives you the flexibility to do just that. Biotech's trusted plate readers deliver precise and reliable results for endpoint and kinetic assays. For 3D confocal imaging, the Cytation 10 captures high-resolution images if you're looking at complex structures such as spheroids or organoids, or if you're looking to monitor real-time cell behavior like proliferation, invasion, or cytotoxicity, then the Xcelligence instruments provides consistent label-free analysis. And for those that are looking to expand their flow cytometry capabilities, Agilent offers the NovaCyte traditional and spectral flow cytometers. In today's webinar, Dr. Burgener will be sharing her experience with the Cytation 5. The Cytation 5 Cell Imaging Multimode Reader is ideal for labs that need both automated microscopy and multi-mode plate reading capabilities in the one powerful instrument. Dr. Sabrina Sofia Burgener is the Deputy Lab Head of the Inflammasome Group, led by Professor Kate Schroeder at the Institute of Molecular Biosciences at the University of Queensland. Her research focuses on understanding how inflammasome signalling is regulated from the cellular level right through to whole organism response, and how this contributes to disease. When I met with Sabrina a few weeks ago to prepare for today's webinar, I was actually genuinely inspired by her commitment to making sure discovery science translates into clinically relevant outcomes, especially for patients that are suffering from rare and chronic disease. I have no doubt you'll find her presentation today just as inspiring. So over to you, Sabrina.
Thank you so much, Julie. And I think I will share my slides. Perfect. I hope you can all see them. And I really would like to thank Millennium Science and especially Julie for the opportunity that I can present today what we're using the Cytation for to actually really delve into inflammasome signaling. And before we start, I actually would like quickly to acknowledge the traditional owners and their custodian of the land that we meet today. So for me, that's here in Brisbane. So I really would like to acknowledge the Turrbal and Urungal people and their land and pay my respect to their ancestor and their descendants who continue culture and spiritual the connection to country. I recognize their value contribution to the Australian and also to the global society. So in the Inflammasome lab, we are really quite interesting on the inflammasome. So I think that's the name where it comes from. But more important is actually the role of an inflammasome in inflammation. And I really like that schematic view that I will show you in a minute, that I got introduced many, many years ago and actually drives my fascination on inflammation. And basically, inflammation is nothing else than characterized by heat. So we all know if we have an infection or even a mossy bite, it actually reacts in heat and then it gets red. And I think that's really science that we can all observe on ourselves, how inflammation plays a role in our body. But more important, it actually leads to a swelling, which then opens your blood vessel and often is considered like with incoming of fluids and all of that. And that actually also results in pain. So inflammation can be extremely painful through this signs, and with that also has an impact that it actually leads to the loss of function. And an inflammasome is actually nothing else than a complex in the cytoplasm of our innate immune cells that then activate inflammation. So how is this inflammasome actually working? So as I said, it's a complex, so it's more than just one protein that plays a role, and it's highly effective in our innate immune cells. So what we will see here is actually a little movie on how an inflammasome works. So if we dive into a cell, for example, a macrophage, which is an innate immune cell, we have NLRP3, which is one of the most well-known inflammasome. So NLRP3 is a sensor that can sense an invading pathogen or cellular stress. And normally it's in this inactive form, so it's really close. But once it senses this disrupted hemostasis, it actually opens up. And this open up allows that several NLRP3 molecules can oligomerize. And this oligomerization leads to this kind of a wheel-like or a flower-like structure that then allows that other molecules can get recruited. And one of these is, for example, ASC. So ASC can interact with this red part of NLRP3 to be recruited to it and then also oligomerize and forms a so-called ASC speck. And this you will hear me later on to refer quite often, because I will show you how we can measure this ASC speck. So once we have this formation of this ASC speck, what actually happens is it allows the recruitment of caspase-1 to the inflammasome. So caspase-1 is a protease, and it actually gets recruited to it and then can cleave cytokines. What is special is actually that caspase-1 itself activates it on the inflammasome to be active. So there is an activation step involved. And then caspase-1 can cleave, for example, IL-1 beta and IL-18 in their mature bioactive forms. But caspase-1 can also cleave gasdermin D in its so-called N-terminal fragment. Now, once caspase-1 has cleaved all these enzymes or its substrates, what actually happens is caspase-1 cleaves itself to terminate its activity and get dissociated from the inflammasome to inactivate this complex. Now, what happened actually is that this N-terminal fragment of gasdermin D can translocate to the plasma membrane and form pores to then activate NINJ1, who leads to the plasma membrane rupture and the cell dies. At the same time, it also allows the secretion of this cleaved IL-1 beta and IL-18 to the secretion to recruit other immune cells, for example, into an organ, but also they actually cause fever, which we know is one of the typical hallmarks of inflammation. So that's basically how an inflammasome works. Now, if we go to a bit less fancy version of this schematic, what we actually know from the literature is that to activate an inflammasome, we need a so-called signal 1, which is through a pattern recognition receptor. For example, toll-like receptor 4 that can sense LPS. What then happens is that we have the transcriptional upregulation of NLRP3 and pro-IL-1 beta. Now, importantly, we need actually a second stimuli, which can be a variety of either cellular stress or invading pathogens that then lead to the assembly of this inflammasome complex. So we have then the oligomerization of ASC to form this ASC speck that then allows the recruitment of caspase-1 to then cleave its substrate. And as I said, what can happen is that then gasdermin D translocates to the plasma membrane, forming pores, activate Ning1, leading to cell death that we call pyroptosis. The reason why we call it pyroptosis is it's a fiery cell death because of the release of the cytokines, especially IL-1 beta, who is the most potent inducer of fever. So what I would like to show you today is 3 different ways how we assess this whole pathway to see if an inflammasome is formed, or especially an NLRP3 inflammasome. So the first one that I mentioned before is this ASC speck formation. So how we actually can assess if an ASC speck forms in a cell. The second part will be about pyroptosis. So can we measure cell death in cells that are inflammasome activated? And the third one is kind of a combination of everything to see how is this actually important in infection. So how this really helps us to protect us from an infection. So if we have a look to ASC speck, so the easiest form of looking at an ASC speck is actually to use quite a simplified system. In our case, we often use a HEC293 T cell system where we stably express ASC coupled to GFP. This then allows us that we can transfect these cells with a construct or a plasmid that has NLRP3 tagged with mCherry. Now, NLRP3 can either be normal NLRP3, or we actually can use NLRP3 that has mutations that we even see in patients. So this really allows us also to mimic what is actually going on in patients. Now, how we usually assess then this ASC speck formation is by using a flow cytometer. So in normal cells, what we have is actually ASC is quite even distributed in the cytoplasm. So we see just overall GFP expression. So if you run this through a flow cytometer and knowing how like the laser works with the height versus the width, what this actually will give you is quite a weak ASC pulse because the GFP is even distributed through the cell. However, once a cell forms an ASC speck, what actually happens is that all the ASC condensates to one central bright spot. And this you can detect by a very strong ASC pulse. And how this looks like if you analyze your data is actually something like this. So we can have all our cells, gate on live cells, and then actually what we can do is here, we see the mCherry expression, which is the amount of NLRP3 that we transfected into these HEC cells. And what we see is that it's actually quite a broad spectrum of expression. And what we know is that if you have high expression of NLRP3, that can spontaneously aggregate. So what you can actually do is you can gate on a moderate or even on a very low expression level and then see how much specks are formed in this context. And if we can see here, for example, if you use a control, which is just mCherry without NLRP3, we see no speck formation. Whereas if we add NLRP3, we actually nicely see this speck. And this we can quantify with something like that, the percentage of specks versus empty vector, and we see massive upregulation of speck formation, or we can also look at that over the expression profile of NLRP3 and then see also the percentage of specks. Now, usually we run this assay as one individual time point. So usually in the lab, we transfect the cells with NLRP3, and then the next day we run them through the flow cytometer, which means A, you only measure an endpoint, and B, it's not very high throughput. And it will not tell you when the speck actually occurs because you just have one snapshot. So we were thinking, can we actually use the citation to get a more kinetic and visual information of this assay? And basically, we did the same layout as we would do for the flow cytometer. So we used our Ask-GFP cells, transfected them with a plasmid containing mCherry expressing NLRP3, added Höchst dye to actually stain the nuclei to know where are the cells, and then straight after the transfection, put them into the Cytation. So not waiting until there might be a speck formed or not, but immediately after transfection to follow up the cells to see kinetically when is the speck formed. So what you will see is in blue, you will see Höchst, which is a nuclease dye. In red, you will see NLRP3, and in green, you will see Ask. And what is really interesting, what you will see is that we see again the same thing that we saw in this schematic view, that GFP is actually evenly distributed in the cytoplasm. But as soon as you will see a red signal coming up, which is the expression of NLRP3, you will see this condensation into this Ask speck. And that's actually how it looks. So what we could actually see with the Cytation is that we have this appearance of this red NLRP3 expressing cells. And as soon as they express NLRP3, the GFP actually condensates and forms this speck. And we can measure this kinetically. So to quantify that, we can actually use similar readouts as we did with the flow cytometer, where we can look at the expression level of mCherry. So we can see that we have, again, high expressing cells versus intermediate versus low expressing cells. And then within these, we can again measure the percentage of Ask specks that are formed over time. So usually with the flow cytometer, we have somewhere a time point on that side of the axis. But what we actually already see is that already after 4.5 hours, we saw the first appearance of Ask specks. So actually the Citations allows us a way more dynamic form or measurement of these Ask specks, and that they appear way earlier than what we have seen before. So the next assay, which we were actually quite interesting, was on cell death, because cell death is one of the major readouts of an NLRP3 inflammasome activation. So the gold standard assay in the field is actually LDH release. So once the cell dies or the plasma membrane is ruptured, LDH is actually released into the supernatant. And then you can collect your supernatant, you add a substrate. What happened is it converts and it makes a color change, and you can use a simple plate reader, measure absorbance, and then you can calculate how much LDH was released. And with that, you can have kind of correlate the cytotoxicity and the cell death that occurs. And usually you have something like that. So you can run multiple plates. In each plate, you can run either a dose curve of your agonist that I will show you in a minute, or you can run your inhibitors. So we try to design different inflammasome inhibitors. So you try to do an IC50 curve, and then you look at that over time. And it then gives you data something like this. So where you see the percentage of cell death, which is like the release of LDH compared to a positive control, and you get an information something like that. So here I use nigericin, which is the most potent NLRP3 activator, and we had a plate after half an hour, and 4 hours using different doses. And what we see is, yes, there is some cell death going on after half an hour, which is dose dependent, and it increases after an hour and looks like stable after 4 hours. However, it is not giving us any correlation between each individual cell, because we have them on different plates and they might react differently, and something happened when you stimulate them or not. So you lose this kinetic information. And in addition, you don't know what is going on basically the first half an hour, except if you would add an additional plate and add a plate at 20 minutes, 10 minutes, 5 minutes, which can be very labor intensive. So we ask simply, could we do a similar approach with the Ask spec, have everything in one plate? As soon as we add the agonist, actually read the plate to see, do we see the occurrence of cell death? And this is what you will see here. So we have Cytox and propydium iodide. These are two dyes that are well known that get integrated once the cell dies. And we can see that we have the nice appearance of green and red cells. And then basically we can quantify that. So that's exactly the same example as I showed you before with the LDH data, where we used a dose curve of nigericin over time. And basically that was in one plate. So instead of having like 3 plates and seeding out a lot of cells, it actually allowed us to use one plate and reduce the number of cells. What is also more important is actually the time points that we can measure that. So in that assay, because we were really interested to capture the cell death as early as possible, we could actually image each well every minute. So that allowed us to have a way more detailed kinetic analysis of cell death going on, for example, here. And we see that we actually already had the appearance already after like 17 minutes. So, and usually the earliest time point we did was half an hour. Importantly, this also allows us to screen like inhibitors or other agonists in a way more high throughput measurement than what we have done before. So now I really would like to shift and go to an infection. So we know that the NLRP3 inflammasome is highly important to protect us from infection, especially from infection from bacteria, viruses, but also from fungus. So the WHO has a list of critical fungus to human health. So these are fungal infections that actually showed a high increase in prevalence and disease severity over the last couple of years, mainly due to the resistance of current antifungals. 2 of them, which are in the critical group, are actually Candida aureus and Candida albicans. And they're quite interesting to us because these solely rely on NLRP3 to be activated. So our immune system, only NLRP3 can actually help us to clear this infection. Now, data showed that we actually, if you have mice that you infect with Candida albicans via intravenous infection route, that these mice start to die. And that actually gets even worse if you knock out NLRP3, really indicating the importance of NLRP3 in protecting us against Candida albicans infection. So we were wondering, can we use a more simple approach in vitro to actually look how does Candida activate the NLRP3 inflammasome? Now, when we infected macrophage with Candida, and we used a reference strain from Candida albicans, which is SC5314, we can see that Candida can induce a cytokine response. So we see this release of these pro-inflammatory cytokines, for example, IL-1 beta. And we also see that it can induce cell death. However, the magnitude is clearly way lower than our favorite agonist nigericin, which is just so potent. However, that's still really relevant in a disease context. Now, we miss one major part, and that is actually the morphology of Candida. So we know that normally Candida albicans is a commensal in our mucosa. It sits with us in our mouth, in our gut, intestine, mucosal, and it's normally in a yeast form, and we do not have any disease with it. However, once we're immune compromised, or in a situation where we have, for example, an HIV infection, Candida actually can cause a huge problem to our health and gets systemically or infects us systemically. And we know that this has to do with a morphological change of Candida from this yeast form. So we really wanted to look at that in a more physiological way and to look at, do we see actually a switch in the morphology of Candida once we infect macrophage? And that's exactly what I will show you. So what we have is again Cytox in green to show does it induce cell death. And in blue, we have a dye called cacofluor white that actually only stains hyphal formation of Candida once it escapes a macrophage. And that's how it looks. So if you infect these cells, we see that we have the slow appearance of dead cells, but importantly, over time, we see how Candida forms this hyphal network on macrophage. And this we could only do by measuring it with a live cell imaging tool. And then we can again quantify that. So here, for example, we can look at the intensity of coccophloor white over time. So we see that like these hyphae are formed over time, and like we talk here about hours. And then we can also follow the cell death pattern. So for example, here we used 2 different doses of Candida. So we had like a low dose, a so-called MOI of 0.3 versus a MOI of 2. And we can really see how the amount of Candida can induce way more cell death compared to, for example, just uninfected macrophage. So as a conclusion, I think what the citation really allows us is kinetically look at an inflammasome signaling. And I think it really allows us to uncover novel and probably also so far missed features on how an inflammasome not only assembles, but then also helps us to prevent an infection. And hopefully we can also translate that into a disease setting where we can characterize what is going on in disease. And with that, I actually really would like to thank all the people involved. So a huge thank to the whole Inflammasome lab, to Kate, who runs the whole group, and to Jared and Byron, who are my PhD students, who really kindly provided me the data that I can share it with you today, and for you for your attention, and I'm happy to take questions.
Thank you so much, Sabrina. That presentation was outstanding. Now we'll open up the microphone to any questions. If you do have one, please raise your hand.
Julie, we do have some questions in the chat. First one from Maha is asking if we could share the link for the video of the Inflammasome recruit.
So you mean the first video that I showed you? I think
Yeah.
that's actually uploaded on YouTube. So, and otherwise, I think it should be on our homepage. So if you go to inflammasome.com, you should also find it there.
Maha also had an extra question asking, how can we use SPECK imaging if we use primary sample monocytes from patients since they don't have fluorophore tagging?
Yes. So it depends on what assays you would like to look at. So we use human-derived macrophage. So we get from healthy donors, we get from the Red Cross, we get blood, and then we isolate either the monocytes or actually we differentiate them into macrophage. And normally it takes like 7 to 8 days for them to differentiate. And actually around day 5 to 6, you can actually genetic manipulate them. So we often transfect either, you can transfect mRNAs into it. So that actually works quite well. You can also use stealth RNAi to knock down things. So it's actually possible to genetically manipulate them and also look at things like that. Or you actually can activate an inflammasome and then fix the cells and then stain for ASK specs, classically through IF.
Okay, thank you. I do see Kerry has raised her hand to ask a question, so I'll take her off mute, and then we have a couple more questions in the chat. Kerry, you should now be able to ask your question.
Yeah, I think. Hi, Sabrina. Kerry Sandman from the Westmead Institute in Sydney. I wanted to ask you if you, when you're looking in different cell types, do you see different numbers of ASK specs?
Yes.
Yeah, is it multiple ASK specs? That's okay in some cell types?
So macrophages are, I would call them easiest because there's 1 speck per cell.
Right.
So it's, I think, very well described, and I think that's a very typical NLRP3 response. So what I have not really went into is that we have more than one inflammasome. So we have actually 9 different inflammasomes, and depending on your cell type, you have a different inflammasome or a preference for other inflammasomes. So for example, epithelia cells in your skin or in your lung highly express NLRP1 or a caspase-4 inflammasome. And there, I think there is emerging data that there can be more than one ASC speck in a cell.
Okay, so we've definitely seen multiple specks in our primary lymph node macrophages compared to MDMs, where we'd see single specks. Is that so? Do you think that's more likely to be multiple types of inflammasomes rather than multiple, for example, NLRP3 specks?
No, I think it's still probably through one inflammasome. Okay. I don't know. I would probably think it's still through one inflammasome, but forming more than one ASC speck.
Okay.
So, but I knew that the data, for example, with NLRP1 is all, I think, still unpublished. So it's just a field that starts to emerge because over the couples of years, everyone focused on NLRP3 because it's the easiest to characterize, especially using nigericin, ATP, disruption of potassium efflux or mitochondria damage. It's quite easily activated compared to others where we first needed to know what's actually the agonists. So but happy to have a chat further, Kerry.
Thank you. And just also quickly on that, do we know if the downstream steps in the signaling cascade are the same regardless of the inflammasome sensor?
Yes. So caspase-1 is the central protease in the canonical inflammasome. So we always know that caspase-1 gets activated, except in human if you have caspase-4 activation, and in mouse it's caspase-11. So that's the so-called non-canonical inflammasomes. But in a canonical inflammasome where you either have NLRP1, NLRP3, NLRC4, you always get caspase-1 activation. So that's the central protease that then is responsible to cleave the cytokines. Now, IL-1 is IL-1 beta is highly expressed in immune cells. So there you will have a preference that then caspase-1 cleaves IL-1 beta. Whereas in epithelial cells, we know that, for example, IL-18 is more expressed. So there you probably will have a more IL-18 signature on the cytokine levels. And then gasdermin D is actually quite ubiquitously expressed. So you likely will get gasdermin D cleavage and then cell death. So we know that the downstream pathways are actually quite highly conserved compared to the sensors.
Great. Thank you very much.
Welcome.
Thank you so much for that question, Kerry. We probably have time for one more? So David, I think you said there was someone else who had a question.
There are 3 more questions in the chat. We'll go with Sifan because they're next in queue.
Sure.
So they are asking, can cell death and ASC specs formation be observed together?
Yes, and yes would be the short, simple answer. No is the complicated answer by the limitations of the color we can use at the moment on our Cytation machine. So the Cytation 5 that we bought has actually 4 LED cubes. So, and with having asked in GFP channel, then mCherry for NLRP3, Höchst in blue, and then Brightfield to actually look at the cells, we actually have that completed. It is possible if you will use one of the channels. So if you would give up, for example, Höchst and say you would use that as a cell death marker, you can absolutely look at that simultaneously. So that would be no problem. But how we have it set up in our system, and we actually cover all the 4 channels with that. But you could also say you will not look at Brightfield and there use a different LED cube and look there on a cell death marker. So yes, short answer, yes. However, it needs a little bit of adjustment to the assay format.
Well, thanks, Sabrina. Maybe we'll take those last two questions in the chat, David.
Sure. So Hong Tree Tran is asking, have you also tried the IncuCyte, and is the Citation 5 more advantageous?
Yeah, so I thought that will be the first question that comes. Yes, we did. So we did a one-to-one parallel comparison. In our view, for our assays that we thought we need, the advantage of the Citation actually was the quicker readout. So the IncuCyte has other benefits, which I think, depending on what you run, will be highly beneficial. For us, it was about timing. So how quickly reads, if you have a full 96 well plate, how quickly goes it from A1 to H12 and go back without needing a break or a pause on heating or then causing stress to the cells. So that was for us the reason why we acquired the Citation.
Okay. And last question I see is from Jack Rivers 40. How do you distinguish between live/dead in flow if GSDM-D is activated?
Not 100% sure what you exactly mean, Jack. So what we have done is, for example, using gasdermin D knockout mice, and then, oh yeah, Jack raised his hand. Maybe you can.
Yeah, Jack, I'll just take you off mute so you can have a chat. Yep, go ahead. You are still muted.
Can you hear me now?
Yes.
Yep, there you go.
Oh, great. So I was, great talk, by the way. I was just asking, yeah, if you're going to use flow cytometry, and I noticed that in your transfection assays, where you transfect in the M-Cherry NLRP3, your selection criteria, you were selecting for singlets, and then you were selecting for live dead. But I was worried that gasdermin D might be activated, given that ASC and NLRP3 are activated, and therefore you might get pores in your membrane, and therefore all the cells that have active specks and active inflammasomes would appear as dead and be selected out.
Yeah, so that's why we use a hex system, because they don't have any of that in. So
so they don't have gasdermin D, the kidney cells?
Yeah, so that's why
nice.
that's the beauty on the hex system, that they basically like almost every inflammasome component, and then you specifically just bring back what you need and then can look at that. So that's different if you would look at the epithelial cell or a macrophage where everything is there, then yes, you actually would have that bias of selecting things out. So that's actually the beauty on the hex system. So, and for cell death, what we normally use there as then controls is then, for example, gasdermin D knockout mice. Then can test if an agonist is specifically leading to NLRP3 activation through then leading cell death through gasdermin D by doing the same in a knockout system. So, but for the ASC system with the hex cells, there the beauty is that they don't have gasdermin D, so we don't need to worry about that.
Nice. Thank you.
Welcome.
Thank you so much, everybody, for those engaging questions, and also thank you, Sabrina, for that amazing presentation. I have a whole heap of questions that I do need to ask you, but I can do them offline. So if you liked what you heard in today's presentation and you want to get in touch with us, please scan the QR code. If you'd like to have a chat with our team about obviously the Citation or any other application that you're working on and some of the instruments that you've seen today. So get in touch with that QR code. So thanks, everyone.
Thank you, everyone.